Good afternoon. Today is Friday, October 2nd. I'm going to be talking about the Chemistry of Life lab about water properties. So for you folks that are remote this year, I'm going to sort of walk you through our stations that we have done in person so you can participate at least on some level in this activity about water properties. So you're going to want to go to Google Classroom. I'm going to share my screen here. And in Google Classroom, the CL3 properties of water activity is what we're after. And there's a Google Doc in there that you can open up. And you can either print it and do this by hand and then take a photo, or you can go directly in the document. All right, so right here, I would read through the introductory paragraph, but right here it talks about what you're going to do. You're going to record a sketch or take a picture of each activity. So you might need to do a screenshot, stop the film and do a screenshot if you don't do a sketch. You're going to write down careful observations about what you see. And then you're going to explain these observations based on the properties of water listed, which I'll walk through in a moment. And then the four of you are going to coordinate putting together a Google slide presentation about the lab following these guidelines. Ultimately, you're going to make 13 slides based on the 13 stations. You guys need to divide that up between the four of you. And somebody make a Google slide doc and share that with everybody else. And then each of you will submit the final document through Google Classroom. You will only be scored on the slides you were supposed to create. So these are the stations. The stations are listed right down here. This is a short description of the activity that is going on at that station. And these are the key terms you need to be able to apply to what you're seeing at the station. These are all terms related to the properties of water. So explain this activity using the term polarity. So polarity applies to every single one of these situations, plus the additional terms that are listed for each activity. This sheet is the same layout, same terminology. There's just a space for you to put notes, comments, etc. Eventually, you will submit this note page, and you will submit your Google Slides for this lab. So I suggest you've done the previous stuff. You've watched the videos, you've done the quiz, you've done the Mastering Bio on Water Properties. This will make a lot more sense if you've done that. So we're going to get up and we're going to move. And I'm going to go station by station. So let's go over here to station number 1. Okay, so let me come back out of sharing. And station number 1 involves this model kit, which is called the water kit. And I'm going to jump this out. I'm going to move my screen so hopefully you can see this. All right. And so this is what's in the model kit. I've got these these molecules represent water. This represents salt, sodium being the blue ball, chlorine being the green ball, sodium chloride, that's an ionic bond. Ions attracting, sodium's positive, chlorine's negative. And this represents ethanol. This is a non-polar molecule, which means it does not have a distinction in charge. Polar molecules, remember, have a split in charge. Water's polar. It's got a positive, negative region. Non-polar molecules have equal distribution of electrons and no negative, positive regions. Okay? So we've got all of these. So here's a single water molecule. And you could probably figure out what's what. What do you think represents the oxygen? Correct. This larger red structure is the oxygen. Therefore, the two white structures represent the hydrogen, H2O. And in the first part, they're asking how many hydrogen bonds can a single water molecule make? So if I take this water molecule, hydrogen bonds are formed between the hydrogen of one water atom and the oxygen of another water atom. So here comes the hydrogen of one atom connecting to the oxygen of another atom. There's one bond so far. I could make a second bond right here. Is there anywhere else we could put a bond? Yes, we can have a positive hydrogen attached to the negative oxygen. We could have a positive hydrogen connect to the negative oxygen. And so that shows you that water, a single water molecule is capable of making up to 4 hydrogen bonds. It's capable of making up to 4 hydrogen bonds in nature. Okay. Let me just check I'm not missing something there. And then 1B says, using the model kit, explain the interaction of water with the model of methane. So we will do that. Whoops, I hit something incorrect there. Sorry, 1 second. So now here I've got some water molecules strung together. And I'm going to actually add salt and ethane into this picture, ethanol. So if I take the salt, let's do that first. Salt is polar because it's got a positive and a negative molecule, positive sodium, negative chlorine. Water is polar because it's got partially positive hydrogen, partially negative oxygen. So that when I put this ion into water, let me throw a little bit more water in here. When I put this ion into water, some of the sodium, some of the positive sodium is going to be attracted to the negative oxygen. And that's going to cause this molecule to split. So this positive sodium gets attracted to that negative oxygen. This negative chlorine gets attracted to that positive hydrogen. And voila, the sodium chloride has dissolved, split. It's mixed in with the water. So polar dissolves polar. That's what's happening here. But if I take this ethanol, ethanol has no polarization. There's no difference in the electronegativity between the two atoms of carbon. The electrons are equally shared. There's no partially positive or negative region. So when I put this into the water, there's no interaction. Nothing happens. If I pick up the water, the oil just slides right off. If I try to put it back into the water, pick up the water, the ethanol just slides right off. Non-polar does not mix with polar. There's no charge on this molecule to interact with the charges on this molecule. So that's essentially stations 1A and 1B. So you may want to pause the video for a moment and make sure that you record some of that information down on your paper before moving forward. So go ahead and pause, and I'm going to move to the next station. All right. And I'm going to walk away from the screen just for a minute so I can grab something that I need. Okay, sorry for the delay. So this is station 2. So 2A is asking us to take a couple pennies. Got 2 pennies. And we're going to take some water, just plain water. And I have some water that has soap in it. I'm going to stir this up because otherwise some of the soap is settled out. As I stir that up, I think you can see there's some suds in there. And it's asking me to go through and figure out how many drops I can put onto a penny before it overflows. All right, so I'm going to move my camera down so you can see what I'm doing. Of course, I don't think I can zoom. So I'm just going to start going. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. That wasn't really 27. There was nothing that came out. This will be 27, 28, 29, 30, 31, 32, 33, 34. 34 is where it broke. And I'm supposed to do that 3 times. All right, so that first time took 34. I'm going to dry up the area, dry that penny off. In fact, I'll just switch the penny out and we'll try that again. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. That time it broke at 24. So 34, 24. And again, I'll grab a new penny. You'll see in the directions, you get to do a little stats with this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. I'm probably going a little too fast. I'm going to slow down. 14, 15, 16, 17, 18, 19, 20, 21, 22. That time it broke at 22. So we had a 34, a 24, and a 22. Now we're going to repeat that process using other pennies, and we are going to do soapy water this time. So this has had some detergent, dish soap added to it. I'll just line these pennies up. All right, so now we've got the soapy water. I'm just stirring it around, make sure I've got it ready to go. All right, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Broke at 18. So 18 was the first one. 1, 2, 3, 4. Oops, I'm not going to count that one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. That one broke at 15. So 18 and 15. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. That one broke at 24. All right, so that was that particular test. That was 2A. 2B says to watch a video. If you go back to the document, which I will do, let me find the document. So if you go back to the document right here under 2B, there's a live link. And you can watch this short video to do 2B. So the link is right there directly in the document for 2B. For 2C, I'm grabbing a bowl of water that we have put some pepper in. Yeah, I'll move my screen down. So that's got pepper in it. And I've got a bar of soap. If I put my finger in here right now, you can see nothing really happens too much. If I take the soap, I'm just going to get a little bit wet. And I rub my finger on the soap, watch what happens. Hopefully that comes through on the camera. All the pepper has moved to the edges and the center cleared out. If I remix that, watch again. If I remix that, stick my finger in, boom. All the pepper goes to the sides, the center clears. So that is 2C. Oh, just made a mess. Okay, nice job by me. I'm more worried about my computer than anything else. Okay. So that's number 2. So let's go over to number 3. Again, you might want to pause the video so you can take notes at any point. I'm just going to keep going, but feel free to stop at any point. So 3A says, observe a beaker of colored water and describe what you see. So here's the beaker of colored water. We've got some green onions in here, and when I pull the green onions out, you can see. What do you see? And I'll take a couple and put them closer. So here's a green onion that's been sitting in that food coloring. Oops. Try and show you another one in case there's anything that might show up different. And so that's more of an observation one. Okay. And then we have another one. So then this gets into 3B, and this is about capillary action. So I have some paper towel. Let me move this seat up. And I've been asked to make a sheet of paper towel that is 2 by 10 centimeters. So that's 2 by 10. And so all I did was cut out a piece of paper towel that's approximately 2 centimeters by 10 centimeters. I'm taking a glass rod and I'm going to tape this onto the glass rod. And then I'm going to take this dry piece of paper towel and I'm going to put it in the beaker until it just touches the water. And as I do that, I'm already losing it. Hopefully you can see if you follow my finger, the water is slowly climbing up the paper towel. So I don't know how visual that is on your screen. If I try this a separate way, let's try this. This might help a little bit. So this is going to be a rough cut this time. So same idea, I'm just going to wrap this piece of paper towel And I'm just going to pour off some of this red water and see if that will help this work any better. Right, so just in case you didn't see that one, let me try this with the red water. And I think you can see what's happening a lot easier with the dye in the water. If you're here in person, it's easier to see. There's actually a clear band ahead of the red dye. I don't know how far the red dye will go up, but the water itself is continuing to climb in an upward direction up the paper towel. And that is the observation for 3B. Now moving on to station 4. So in station 4, I have a set, everything's backwards on the camera, so I got to make sure I'm doing this right. I've got a set of test tubes. I'm going to just put some water in and clean these out. So I just got some test tubes. I'm taking my brush and cleaning them out. Okay. And then I'm supposed to add, let's see, pinch of salt in one, and I'm going to put a pinch of sugar in the other. I'm going to add 10 mils of water. So here I have some salt. And I will, just to make it hopefully more visible, put a couple pinches. Wipe off my hand. And I will put a couple pinches of sugar. Okay. And so now I'm going to add some water to each one. So this is the salt. I'm going to put approximately 10 mils of water in. All right. And then I'm supposed to just vigorously roll it around to mix it. And what I can see, yeah, I'm not sure how well you can see that. I'm going to flip it a little bit. There's no salt crystals at the bottom. There's no noticeable crystals in the solution. The salt has completely dissolved in the water. Now, if I repeat that with the sugar, so if I do that with the sugar, I'm going to see that there's still sugar crystals in the bottom. And if I keep trying to mix it, it does not 100% dissolve. Sugar is remaining intact in the presence of the water. Okay, so that takes care of station 4. So the demos, the activities are very simple, but it's the applying the knowledge that's the important part here. So let me move on to number 5. Number 5. So I've got 2 containers of water. So I'm going to add some water. Let's see how much do I need. I want 2 to 3 mils of water. And in one of them, I'm going to, oh, I'm jumping ahead of myself. All right, so sorry, one of them gets water, not both of them. And I'm going to put a drop of oil. So here I've got my cup of water, H2O. And here I have my oil, and I'm going to put some drops of oil into this container. So I put a few things in there, and if I see the oil, I can see the drop is just sitting there. And if I move it around, it sort of stays as one big swirl. I don't know how well you can see that, but it's quite obvious that the oil does not mix with the water. Whereas if I take a container with alcohol and add a little bit of alcohol, and I put my oil into the alcohol, right? I don't know if you can see those 3 little drops, and I start moving it around, I can see that it breaks up into smaller chunks of liquid, smaller chunks of lipids, I'm sorry. So the alcohol is able to get in between the lipids and break it into smaller particles of lipids. So in a sense, the lipids are dissolving in the alcohol, whereas in my water example, the oil doesn't separate. In my water example, the oil stays together, separate from the water. I don't know how well those two particularly showed up on the screen. Probably not very well. And then finally, station 6. In station 6, we used some hot plates, and we had 2 containers. We had a container of just plain water, and we had a container of oil. We took the initial temperatures. You'll want to write these down. The initial temperature of the water was 20 degrees Celsius. The initial temperature of the oil was 21 degrees Celsius. We then placed them both on their own hot plates. So we put water on one hot plate. We put oil on the other hot plate. Bear with me. They were both started at the same time. They were both set on the same temperature. We let them go for 3 minutes. And then after 3 minutes, we inserted thermometers and got the temperatures. So water started at 20, it ended at 60. In 3 minutes, it went from 20 degrees Celsius to 60 degrees Celsius. The oil in 3 minutes went from 21 degrees Celsius to 100 degrees Celsius. So a big shift in temperature, oil shifting much greater than water. And then the last station, we took alcohol and we took water. Okay. Took alcohol and we took water. And we put a drop of alcohol on the counter. And we put, somewhat of a distance away, we put some water. Should make it about the same size. All right. Then we took our fingers and spread it out. And we waited to see how long it took for each one to evaporate. And we have a timer going. And so you can see the alcohol versus the water. So we're waiting for paint to dry. And if I just spread this a little bit more and spread this one a little bit more. I should notice that the alcohol is disappearing much quicker. We're just waiting. And again, I put down way more than I should have. But if we take times from the class, on average, the alcohol was evaporating within about 45 seconds. The water was evaporating within about 4 to 5 minutes. So typically the alcohol goes about 45 seconds. The water goes about 4 to 5 minutes. Difference in evaporation rates. Okay. So that is the 6 stations. I think that should give you enough information to make good notes and to be able to work this out. So again, you'll complete your note sheets. The note sheets should be very thorough. You need to apply the terms that are provided to what was going on with what you observed. You might want to include screenshots, sketches, and then divide those slides 1A through 6B. Divide these 13 stations, so that's 13 slides, up. So each of you is doing approximately 4 each, and you will be graded on the ones you do. Everything goes into one Google Doc. Everybody submits the same Google Doc. Okay. Remember, slides should be short and simple. Images, short statements. Don't give me slides that are dense with text, like 3 paragraphs at a font of 10. I'd rather have you make 2 or 3 slides to explain a concept than to cram everything into one slide where it's very cluttered. Okay? And so those things should be taken care of based on the schedule on Google Classroom. Shortly, I'll be sharing also a video on the pH and buffer lab. Enjoy the weekend.